Hey Bio30s, it's Manchak here with another video lesson for you, and exciting, we are going to wrap up the second part of Unit C, which is our unit that's gone through cell division. We've talked about classical genetics, so we're going to wrap up classical genetics in this video, and then I'm going to introduce to you where we're moving on to next, which is more of the modern kind of genetic stuff that we're looking at the DNA molecule itself and how it works. So before we get into that, I just want to mention to you really quickly that concepts C8 and C9 is what we're working on. This video is going to cover concept C8 and wrap up our classical genetic stuff and transition us into our what we call molecular genetics stuff, which is going to deal with DNA replication, protein synthesis, those kinds of things. So this is a transition video and a transition concept. Hopefully it won't be too terribly long, but watch out. This is the first video of two that you're going to have for concepts C8 and C9. So just really quickly, I'm going to tab over to here and just let you guys know what concept C8 is all about. So remember, we've gone through all of these concepts already. The first three here were related to mitosis and meiosis and how they work. We've then moved through different patterns of inheritance through C4 through C6. Then we talked about gene linkage and crossing over in the last lesson as well. Now we're moving on to concept C8, and that's what we're covering right now. Basically, concept C8 is a history lesson. We are going to explore how people like Franklin and Watson and Crick, as well as others before them, came up with DNA as being the hereditary material and what DNA looks like. Then that transitions us into concept C9. And C9 is again going to look at the DNA molecule itself. What does it look like? What is it composed of? How does it work? And then how do we set ourselves up for cell division properly? In other words, how do we get enough DNA in the cell, replicate or duplicate the DNA in such a way as to make sure that when we're done, we've got the right amount of DNA in each one of our cells. So that's what we're working on. C8 is this video, C9 will be the next video, and we're nearly done Unit C, believe it or not. Couple of things to say then about this. C8 is going to be, like I said, a history lesson, and it's going to basically track our path through figuring out that DNA is the hereditary material and what DNA looks like. So we're going to talk about a whole bunch of people's names. So be warned, I'm going to be asking you about people's names and what they discovered and what they were able to figure out. To me, I don't need exact dates as to when this stuff happened, but I would like you to be able to associate that this person discovered this thing, and it happened before this person or people discovered this thing. Does that make sense? So try to build a timeline, but exact dates aren't important to me. C9, I said already, is going to go through the structure of DNA, and we're going to get into what it looks like, talk about things like 5' and 3' ends. We're going to talk about semi-conservative replication and the actual process of replication, including, and these are just my notes to remind me, including things like leading versus lagging strands, enzymes, that kind of stuff. So concept C8, these are your textbook pages, and all of this is posted just above where you found this video in the Google Classroom, so you don't have to scribble it down. I've got a whole whack of videos here. I just started digging and went down a rabbit hole for the last 20 minutes trying to find good stuff, and there's a ton of it out there. There's a couple of really good ones. I like this one for an explanation briefly about what happened before Watson and Crick came on the scene, and that makes no sense right now, but it will in a minute. This is a nice summary of the history and discovery of DNA. You can go check that out. Really, I went back to Amoeba Sisters, and these last three are Amoeba Sisters. They do such an excellent job, and the stuff is bang on with what we have for biology here in Alberta. So I would strongly these two, pardon me, this one I'll talk about in a second. I would strongly recommend watching the whole of this one. It's really good, and it's going to take us actually through a lot of stuff we've talked about already. So it might be a nice little review for you. This mechanisms of DNA is one that I always show in class, and we tend to break it down and how it works. So be aware that I think that's an important one to watch out for. And then lastly, DNA replication is in concept C9, really important They do a great job of breaking this down and how it works, including all of the players involved, all those enzymes that get really confusing. For Concept C8, your only formative work is going to be just doing some questions out of the textbook and maybe taking the time to study that timeline and making sure you understand who these people are and what they did. C9, you'll have some questions out of the textbook and then some LSMs. These are just workbooks or worksheets that came from the textbook folks, and I'll post those in the Google Classroom under worksheets for this unit. And then you'll have your practice quiz. Do your practice quiz first, wait for my feedback, and then move on to your summative quiz. So I think that's about it. Let's move our way into the first video here, or the first Concept C8, and get that done and out of the way, and then we'll move into Concept C9 in the next video. So this section, like I said, C8 is sort of a transition. We're moving away from classical genetics, which is stuff that we can derive with not really knowing about DNA itself and what DNA looks like and how DNA works. So all of it is, it's classical because it was all sort of owes its origins to Mendel, which we talked about way back in Concept C4. And we've built off of Mendel's work through various other people in history so far. But what we really haven't talked about is DNA and what DNA is, what it looks like, and how it works. What we're going to do now here is we're going to figure out how we determined that DNA was the hereditary material, how it was the thing that allowed you to look similar to your parents but not exactly the same, similar to your siblings but not exactly the same, how our genetic information is passed from one generation to the next. And it is a bit of a history lesson. It takes us through time. But what I really want you guys to focus in on are the discoveries that took place during this time, starting back from when Darwin was doing his theories of evolution up to about the 1950s-ish, and see all the little pieces that had to come together for us to figure out that DNA is the hereditary material, and hey, this is how it works. We're still discovering bits and pieces about how it works, what it does, how pieces of DNA interact with each other and other things, how we can modify them and change them. So we're learning all of this, and it's going to take time for us to really figure it out completely if we ever do. But this gives you an idea, the science that had to be done to make this happen. And it's really neat when you start to look at the science that goes on. At least it's fascinating to me, and how one piece led into another, and how maybe two separate pieces came together that somebody built off of those. It's the nature of science. It's really how it works. So let's move on here. We're going to start in the 1860s with a Swiss biochemist named Friedrich Miescher. I don't know if I pronounced that correctly, but that's how I'm going to do it. And if I did it wrong, somebody can come and correct me sometime. What he did, he was working in hospitals, and he during a war, and what he ended up doing is extracting stuff from the bandages of wounded soldiers, and he is extracting viscous white substance out of these bandages, which we should be able to identify as the pus that comes out of the bandages. And the pus, if you remember from Bio 20, is full of white blood cells, and these white blood cells contain nuclei. And he was really curious about what was going on with those nuclei. So he found that in the nuclei there was an acidic portion he called nucleic acid. Nuclei, nucleic, get it? Okay. And then there was an alkaline portion, a protein portion inside of the nucleus as well. So he had this inkling that there's got to be something in the white blood cells, maybe in the nucleus of those white blood cells, that is allowing stuff to be passed from one generation to the next. So he narrowed it down. He said it could be this nucleic acid thing, or it could be this alkaline thing, this protein. So he wasn't sure. He named this substance that he found, that he pulled out of it, and it was composed of phosphorus and a nitrogen material. He called it nuclein, because he found it in the nucleus of the cell. So he took this stuff out of the nucleus. It had an acidic portion, it had an alkaline portion. He called the collective thing together nuclein. Okay, and the nucleic acid he found was later determined to be DNA and RNA. But back then, the technology didn't exist. The knowledge didn't exist for him to maybe be able to figure that out. But he figured that this nuclein that he found had the hereditary material in it. Okay, it contained within it that hereditary material. We're going to fast forward a little bit. I'm going to jump back and forth on the timeline a little bit. We're going to fast forward a little bit to 1943 now, and a Danish biologist named Joachim Hammerling. And he suggested that the nucleus was the location of where the hereditary material would be found. And he did a really, really simple, very elegant experiment that allowed him to determine that. And what he did, and this is just a diagram out of your textbook, and I'll get my photo out of the way here so you guys can see it, is he took this very simple organism here, which is a very simple plant, and I'm completely blanking on the name of it right now, but it's going to come back to me in a moment. And what he did is he took this very simple organism, and he knew that the nucleus was down here at the bottom, and this organism would grow from here up to a cap. And he would, the foot had the nucleus in it. And what he did is he took the cap off of it and he let it grow. And lo and behold, a new cap regrew off of it. He thought, that's pretty cool. I wonder what's going on with that. Then, to alter his experiment, he said, okay, well, maybe there's something down here in the foot where the nucleus is. So he went and he took the nucleus off. He cut the foot of this structure off, and nothing regrew. And so he went from there and he said, okay, I'm going to make an educated guess based on my experimentation, and I'm going to say there's something up with the nucleus that contains the information that's necessary to regrow the cap. It contains the information that's necessary for this organism to do what it's supposed to do. He pointed to the nucleus and said, in there is where the hereditary material is. And so Hammerling said we should be looking in the nucleus. What we knew at that point is that proteins and DNA are present in the nucleus. We didn't call it DNA at that time, but there was proteins and something else in very large quantities. But what was really, really interesting is the DNA was thought to be too simple to be something that could be the blueprint for an entire organism and be the blueprint for the hereditary material. That when they broke it down and separated out the DNA and looked at what made it up, they found that it really had 4 variations. It had an A, a T, a C, and a G, an adenine, a thymine, a cytosine, and a guanine. It had those 4 things. Whereas proteins were made up of 20 different kinds of amino acids in ridiculous combinations. Protein had to be the hereditary material because it was so much more complex than the DNA was. So we've narrowed it down to the nucleus, and we said, okay, we got protein, we got DNA in here, or this thing that we will call DNA. But I think that, you know, the scientists at that time were thinking, well, DNA is too simple. It can't be doing anything important, so it's got to be the protein that we're working with. Right now, I'd like you to hit pause, right? This is something that we would do in the classroom. I'd like you to hit pause, and I'd like you to go to your textbook, open up and read 2 pages in your textbook. I know you can do it. And look at what Griffiths did in his experiment and what they concluded based on their experiment. So hit pause right now. I'm going to do the same, pull out my textbook and have a look at that. Hint, I've already done it. And then we'll talk about it in just a second. Okay, so how did that work for you? What did you figure out? What did they conclude? Well, let's sort of just take a step. And I got the textbook out in front of me here, and I'm just I'm just going to refer back to it from time to time because I don't have it all memorized. But I think things should make sense, right? There's 2 forms of this. There's the S form, which is sort of an activated, a form that'll actually kill you through pneumonia, and then the R form doesn't kill you. It's harmless because it doesn't have that coating on the outside that can kill you. So what ended up happening is it's pretty simple. Mouse A was injected with the S pneumonia, and it died. Mouse B was injected with the R pneumonia. It didn't die. Okay, and then they sacrificed the animals. They did autopsies, and they found that the S cells were in A and the R cells were in B. So we can conclude S cell kills, R cell doesn't. When we move on to mouse C continued to live. I'm looking at the evidence section on 645. Mouse C was sacrificed. We did an autopsy, and no living S cells were found. So what can we conclude from that? We can conclude that if we heat treat the cells, right, we kill those cells and we inject them into the mouse, nothing happens. Okay, those cells don't work. Now, mouse D is the interesting one. What they did to mouse D, if you remember, is they mixed heat-killed S cells, which we know did not kill mouse C, and they mixed some R cells. And what they did is they injected into that mouse, and that mouse died. How the heck does that work? Well, there's got to be something that transferred from those dead S cells into those R cells and changed those R cells over to actually kill that mouse. So there must have been some sort of a transformation that took place. I think that's pretty interesting, and it's something that definitely I'd like you guys to think about. And the really short 2-minute and 18-second video goes into this a little bit, and I would encourage you to go have a look at it. So here's where we go from now. We've definitely found out that there's something inside that can be transferred. There's something in our cells, whether you're bacterial cells or human cells, that allows us to transfer genetic information from one generation to the next. So following Griffith's work and the work of others, it's decided that proteins were not the material of heredity. We think then by killing those proteins, by heat treating those proteins, that if the proteins were the material that was transferred from the S cells to the R cells in Griffith's work, that doesn't make sense. So there must have been something else inside the nuclei, inside those cells, that allowed for that transfer to take place and allowed for that bacterium to kill those mice. And so American scientists Hershey and Chase conducted experiments using a bacteriophage, a virus basically, that infects a bacterial host. And that's, interestingly, what all viruses tend to do is they tend to inject DNA into a host, and we know this now, they inject DNA into a host, and that host dutifully produces the proteins that are associated with the DNA that was injected, and that's what makes us sick when we get sick with a virus. That's a really pared down version of it. So just so we're clear, a bacteriophage consists of two parts. It consists of DNA and a protein coat over top of it. Okay, proteins contain sulfur but no phosphorus, whereas DNA contains phosphorus but no sulfur. Okay, this is going to be important because what Hershey and Chase did is really, really, really cool stuff. Is they took radioactive forms of sulfur and phosphorus and they tagged the parts of the bacteriophage. So what they did is they grew these bacteriophages, these little viruses, in a scenario where they would have to take up radioactive sulfur and radioactive phosphorus. And then what happens is it was hoped that the radioactive tag then would show up inside of the bacterial cell. So a bacteriophage goes and it shoots at DNA or shoots something inside of a bacteria and infects it. Okay, so what happens is they're hoping that one of these two, hopefully not both, radioactive things, either radioactive sulfur or radioactive phosphorus, would end up inside the bacterium, and they could find the culprit of the thing that is causing that contains the DNA, that contains the hereditary material. So here's how they did it. Is they took a bacteriophage. In this case, this one was tagged with radioactive phosphorus. Okay, and if you forget, radioactive phosphorus, okay, is DNA, right? DNA has phosphorus in it. And so what happened is the bacteriophage would come along, it'd drop off its tagged DNA inside, and we found P32 or radioactive phosphorus inside the cell. And once after that bacteriophage had disappeared. What's interesting after that, I'll move this out of the way, is that they also injected them with the protein case that was covered with radioactive sulfur. And that bacteriophage would come up, it would inject something into the bacterium, and there would be no radioactive sulfur found in the bacterium. So here's what we've got. We've got experiment 1 where infection occurred and radioactive phosphorus was inside the organism. In experiment 2, infection occurred, but we have no radioactive sulfur inside the organism. So what was the thing that was injected inside the bacterium? Had to have been the phosphorus. We know that the DNA had the phosphorus in it, so it must have been the DNA that infected. It must have been the DNA that infected that bacterium. Does that make sense? I hope. So we've got the phosphorus that is inside the DNA that infects the bacterial cell. So just to summarize again, both bacteria got infected, but only one of the radioactive things showed up inside the bacteria. And it was the phosphorus that was attached to the DNA that showed up inside the bacterium. So that pointed the finger to DNA as the hereditary material, as the thing that was passing on the infection from the bacteriophage into the bacterium. So let's move on. So once it was determined then that DNA was the hereditary material, the race was on to discover what DNA actually looked like. So in other words, we need to know, we know it's DNA now. How does it work? What is it? How does it work? What does it do for us? How is all of this making sense? And so the discovery of the structure built on the essential work of somebody named Rosalind Franklin. Okay. And what I want to point out, and I always point this out every year, is Rosalind Franklin was screwed over by the Nobel Prize people. When Watson and Crick, who really created that first 3D structure of DNA, they were the ones that were given the Nobel Prize, and Rosalind Franklin was not given that distinction when the Nobel Prize was first awarded to Watson and Crick, which is a big, and part of it, and a lot of it points to some sexism and the thoughts back in the mid 1900s that women weren't capable to do science and those kinds of things, which we know is to be a complete falsehood. And she was left off, and I believe she was awarded the Nobel Prize posthumously, meaning that she was dead after when she got that award, which was a number of years later after the original award. But Rosalind Franklin used a technique and was essential in sort of showing Watson and Crick how all of this stuff worked. And without her pointing to X-ray diffraction and showing them how it could be used, they would not have constructed that 3D model of DNA that we're comfortable with now. But then Watson and Crick came in and sort of put all the pieces together and constructed the 3D model, taking into account its chemical structure. So they knew at that point that the hereditary material DNA contained a deoxyribose sugar, some phosphate groups, and nitrogen bases. So 3 real components. When we get into C9 in the next video, you'll see what I mean by these. So for now, it probably sounds a little weird. But all 3 of those things were present, and then they had to figure out, okay, well, how are they stuck together? And then how did all of this stuff lead to the 3D structure that allows DNA to do what it's supposed to do? So they eventually found that the shape of DNA was actually based off of, if you imagine a ladder, which has the vertical part that we hold on to on the sides there, and then you got the rungs that come across the ladder, right? The backbone that we have of this is a double-stranded portion, a backbone, so one on either side, of your deoxyribose sugars and your phosphates alternating in that backbone. And then the rungs of the ladder are a nitrogen base, or a series of nitrogen bases that are paired up in the middle. And we'll get to this in C9. If it doesn't make sense right now, my waving around of hands makes no sense to you, that's okay. What that ladder structure, though, is a twisted ladder structure, meaning that if you imagine taking a ladder with your giant strength and twisting that ladder, that is the structure of DNA. And it was Franklin's X-ray diffraction that allowed them to shine the X-ray onto it and see this world structure that led them to imagining that this is what the DNA structure looked like. Okay, so it is made up of a deoxyribose sugar and a phosphate. This is the backbone, and this is kind of twisted 90 degrees. And then we got nitrogen bases. So one of these things would be called a DNA nucleotide. We're going to get into that in concept C9, so don't stress about that too much right now. So the last thing I wanted to mention is this little timeline here that is included in your notes, and that it comes out of your textbook, and it would be worthwhile for you guys to have a look at it and spend some time with it. I don't want you to know everybody on here, and I'm just going to quickly highlight the ones that I would like you to know what they did and who they are. And remember, dates aren't essential. You'll notice here they've just done years. For me, if you can just get them in sequence, that's great. So we talked about Meischer and what he did, and found that there's a non-protein substance from the nucleus in those cells. So there's something that he called nuclein that was really important. Griffith came along in 1928, and remember, I skipped around a little bit, and they did that experiment with the virulent and non-virulent forms of the pneumonia bacteria. And basically what they discovered is that there is something called transformation that takes place, that something can be shot from a dead bacterium into a live bacterium or taken over, and it was definitely not something protein-based. Hammerling came along and discovered, it was an algae that he was doing. I see it didn't even come to me. And he hypothesized that the hereditary material is definitely stored in the nucleus. Then it was Oswald, Avery, and McCarty, which one of the videos talks about, actually, that did the transforming. Don't worry about them. Don't worry about Chargaff just right now. I will ask you about Hershey and Chase and what they did. And they basically suggested that DNA is the hereditary material based on what they figured out with their experiment dealing with those radioactive phosphorus and sulfur. Then Rosalind Franklin came around, and she produced an X-ray diffraction pattern of DNA that suggested a shape of a double helix. And then it was finally Watson and Crick that came along, and they got that structure. They deduced the information using stuff from Chargaff, Franklin, and Wilkins as well. Okay, so really, I want you to do the ones that I've highlighted on here. So Meischer, Griffith, Hammerling, Hershey and Chase, Franklin, Watson and Crick, and be able to tell me what they discovered, and maybe more or less put those in order as best you can. That's it for Concept C8. That's all I have to do. Sorry that took a little bit longer than I expected, but hey, it's a nice little history lesson. What you're going to do now is you've got some questions some questions to work your way through out of the textbook. Please go ahead and do those. There's a couple of videos on the Google Classroom that I'd like you to watch as well. Once you're done all of that, feel free to jump into Concept C9, where we're going to explore the structure of DNA, talk about how it's put together, and then talk about how it replicates. So I think that's about it for now. May the 4th be with you, because that's when I'm recording this, and we will talk very soon in Concept C9.